Welcome to A2 Homeostasis. At AS level, your focus was on isolated cellular processes. Now we shift to complex whole organism coordination. Homeostasis is the maintenance of a relatively constant internal environment, specifically your blood plasma and tissue fluid, despite wild fluctuations outside. Without this dynamic equilibrium, your enzymes would denature and your cells would undergo osmotic lysis.
How is this balance achieved? We will start by looking at the fundamental control theory. The core mechanism is negative feedback. Whenever a variable deviates from its set point, a coordinator triggers effectors to oppose the change and restore balance. We will also look at positive feedback, which amplifies deviations, though this is rare and mostly restricted to action potentials. Crucially, Cambridge no longer tests thermoregulation. Do not waste revision time on sweating or vasodilation. Your mammalian focus is entirely on water and glucose.
We begin our mammalian physiology with the kidney, the master organ of excretion. You will trace the exact pathway of blood and filtrate through the microscopic nephrons. High hydrostatic pressure in the glomerulus forces small molecules into the Bowman's capsule. This brute force first step is ultrafiltration. But we cannot afford to lose all those nutrients. In the proximal convoluted tubule, selective reabsorption uses specialized co-transporter proteins to rescue 100% of the glucose and amino acids back into the blood.
Once the nutrients are safe, the kidney must balance your water levels. This process is called osmoregulation. You will learn how the loop of Henle creates a steep salt gradient in the medulla. Then we will map the exact cell signaling pathway of antidiuretic hormone, or ADH. You will see how ADH binds to the collecting duct, triggering a cascade that inserts aquaporins into the membrane, allowing water to move by osmosis down its water potential gradient.
Next, we tackled the control of blood glucose. You will explore how the pancreas and liver coordinate to ensure your cells always have enough respiratory substrate. We will map the antagonistic actions of insulin and glucagon, and you will need to perfectly distinguish between processes like glycogenolysis and gluconeogenesis. We will then apply these principles to understand the biological basis, causes, and management of both type 1 and type 2 diabetes.
Finally, homeostasis is not limited to animals. Plants must meticulously balance their internal conditions to conserve water. You will examine how guard cells act as independent homeostatic effectors, regulating stomatal apertures through precise ion movements. We will also look at how the stress hormone abscisic acid, or ABA, overrides normal cellular functions to force stomatal closure and prevent dehydration during a drought.
This unit is highly interconnected. Hormonal signaling pathways rely heavily on the receptor principles you learned at AS level, while the movement of water always comes down to water potential. Master the vocabulary early, map out your stimulus to response loops, and you will dominate the paper 4 structured questions. You are ready. Let's begin.